Before we get into gene engineering, I want to give you a quickie review of CRISPR as it naturally occurs in bacteria. As you may recall, we have the immunization phase, which consists of the period where after the bacteriophage has inserted its foreign DNA, a Cas protein comes along and actually cleaves out a short segment of that DNA and actually inserts it in the CRISPR array, which is a location on the bacterial genome. The CRISPR array consists of these repeats and spacers, spacers being the individual unique DNA and repeat DNA being just what it sounds like. So these are transcribed and processed into individual cRNAs, and each cRNA contains a repeat and a spacer. Now, when this comes together with the tracer RNA and complexes with the Cas protein, we have a situation where this individual spacer is going to be able to hybridize with the viral genome when the cell is infected by the same bacteriophage. We call this the interference phase. And since this is specific for this segment of the DNA, it will only be able to defend against this particular bacteriophage. The hybridization is a signal to Cas9 to actually perform cleavage. And it will do exactly that. It will cleave this DNA, and that will effectively take the bacteriophage out of commission, and the bacteria can successfully defend itself now against attack by this particular bacteriophage. The question before us now is, how can we use the naturally occurring CRISPR pathway and take that and make it a system that we can use for the purposes of genetic engineering? The first thing I want to point out is notice the similarity between this circular bacterial chromosome and this circular plasmid here. As you know from what you have learned in this course, we can modify a plasmid and insert segments of DNA that we would like to have inside that plasmid. So if we're going to make CRISPR work in mammalian cells rather than bacterial cells, we need to insert specific segments of DNA that are the components of the CRISPR system. So what are those essential components? Well, those essential components are something that is going to be analogous to the spacer. In other words, something that is going to target a very exact location in, let's say, a particular gene in a mouse chromosome. So that's one thing we need. We need our guide RNA. We need Cas9. And then these blue segments are promoters. Promoters have the job of turning on the transcription of this target sequence DNA of the guide RNA, and of also turning on the transcription and translation of the Cas9 gene. So what can we do with this plasmid? Well, once we've engineered it to our satisfaction, we can then transfect it into a mammalian cell. And by transfection, what I mean is simply getting it to cross the border of the cell membrane into the mammalian cell, and then to start doing its job, which is to replicate, to transcribe, and to translate. So this is the essence of the engineered CRISPR system. Finding a vector, in this case our vector is a plasmid vector, that we can insert the essential components of the CRISPR system, getting that into the mammalian cell, and then having it start manufacturing the CRISPR components that will target a specific location in the mouse genome and make that double-stranded break. That doesn't answer the question of how we can make changes to that DNA, but that will be coming up shortly. This slide recaps what we just spoke about, and this is the idea, the concept of targeting a specific sequence. So if you focus your attention down here, this would be part of a genome of, let's say, the mouse. And this would be double-stranded DNA right here. So once we transfect the mouse cell with the plasmid will have made this construct, as well as having produced this Cas9 protein over here. Now, what you might notice is that the spacer and the repeat and the tracer RNA are now no longer separate. So what researchers did is they simply said, okay, we're going to make this stretch of RNA all one piece. We're going to fuse the tracer RNA to our spacer repeat, and it will do the job in the same manner. And that's true, it actually does. They don't particularly need to be two separate portions of DNA. I mean, of RNA rather. So this now can be called synthetic guide RNA. And notice that it is what is analogous to the spacer in bacteria that hybridizes to the target site in the mouse genome, and then once complexed with the Cas9 protein, it will go ahead, the Cas9 protein will go ahead and do its usual job, which is to cut this DNA into pieces. Now, notice once again that we need the PAM site to make this work. So the target site in the mouse genome actually has to be adjacent to this very short stretch of DNA called the PAM sequence. This is something you should just keep in mind. It is a little bit limiting in terms of what target sites you can use in the mouse genome because you need this PAM site. It is, however, extremely short, so it isn't that hard to find a lot of these PAM sequences scattered around. The next question is, how are we the scientific community, going to alter this DNA that we have targeted once we have cut it? So this is where things get a little bit more complicated and yet still quite doable. Now we get to the really interesting question of how can we use the CRISPR system in a mammalian cell to perform gene editing. In other words, not to just cut the DNA, but to make changes to the DNA. And in order to do this, it's necessary to rely on some of the mammalian cell's own mechanisms for DNA repair. And the two things that we're going to talk about in the context of DNA repair are non-homologous end joining and homology-directed repair. So these are two terms you need to be familiar with. We're going to talk about non-homologous end joining first, which is a kind of DNA repair system. So imagine that you have engineered a CRISPR system, you've put it into the plasmid, you've transfected the cell, and that CRISPR system that you have designed goes in there and it cuts the DNA at a very specific location, the location that you want. Well, now if we just leave things alone, the cell will attempt to repair that double-stranded break. And in doing so, we're going to have something called non-homologous end joining. So the interesting thing about non-homologous end joining is if left to its own devices, it's not terribly precise. So sometimes it will stitch the ends together correctly, sometimes it will not. If it does, let's say, a flawed job of repair, you will end up with something called indels. In stands for insertion and del stands for deletion. So at this site of the double-stranded break, it is very possible that we would get a DNA base or several DNA bases inserted or several DNA bases deleted. These changes could result in a premature stop codon. It could certainly throw off the DNA reading frame so that we would get a protein that is not expressed properly. And the overall effect of this would be basically to knock out the gene, which means in essence to delete the gene, to keep it from functioning, to keep it from making a functioning protein. That was my dog. Now, if we talk about homology-directed repair, This is something that allows us for a more precise kind of gene editing. And for this, you actually need a DNA repair template in addition to the whole CRISPR system that we've talked about previously. We don't have to go into too much detail about, how that can be performed, but know that once you have this break, you basically also provide to the cell some template for repair. This means that the repair job is going to go forward correctly. We're not going to have insertions or deletions or premature stop codons. But what we can have, depending on the template that we provide, is, let's say, a single nucleotide change. Now, as you can imagine, that could be very powerful. A disease like sickle cell anemia is dependent on one nucleotide change. So if we could change that nucleotide back to the gene sequence that is the benign gene sequence, in other words, that doesn't bring on sickle cell anemia, well, then we've done something pretty amazing. Now we're going to look at the three major approaches for gene editing that incorporate either the non-homologous end joining or the homology-directed repair, or even some new directions that CRISPR is going in with respect to things that can be done to the Cas9 protein. So first we're going to focus our attention on the left-hand slide here, the left-hand part of the slide here. This is what we were talking about a moment ago. There's the possibility for non-homologous end joining, which will simply mean that the cell is left to its own devices to repair the DNA. This leads to insertions and deletions and making the gene non-functional. The other possibility is providing some donor DNA or template DNA. This is perhaps a more powerful technique because we have a more precise control over the recombination, the repair of the double-stranded break, and we can actually insert a new sequence if we want to. We can make modifications to individual bases or even more. Now, since the homology-directed repair in combination with the double-stranded break is not always terribly efficient, another possibility that researchers have worked on is to modify the Cas9 so that it only makes single-stranded breaks. Now, if you look carefully at this figure, you'll notice that we have CRISPR system coming in here and hybridizing with a single-stranded break event. And then we have another CRISPR over here and another single-stranded break event. So what does that mean? That means we now have cuts that are staggered. This is actually a better situation for inserting donor DNA. We don't really have to go into why, but it is potentially more efficient. But the goal is exactly the same. We're doing homology-directed repair in order to get some new DNA and to gain control over what changes we make. Now, in the third segment of this slide, researchers are exploring all kinds of ways that Cas9 and the CRISPR system can be used to make changes to how the DNA of interest is actually regulated or controlled in the cell. So let's say we want to have a gene make more of its product, make more RNA. So what happens here is there's an activator actually fused to Cas9, so we get enhanced expression of our target DNA. Another possibility is to modify Cas9 so it's fused to a repressor, and we literally repress expression of our DNA or our gene of interest. And then finally, we could even fuse GFP, which is a fluorescent protein, to the Cas9, and this could help us with the visualization of what's going on at this particular gene location. So The possibilities with CRISPR are, I don't know if they're limitless, but they certainly are enormous. And it's going to be pretty exciting to watch in the next few decades what scientists are going to be able to do in terms of curing disease and other aspects of human biology going forward.